You ever walk through the produce section and notice how all the apples or tomatoes look, well, pretty much the same? That kind of consistency doesn't just happen by accident. It's actually the result of some seriously cool and deliberate science. So today, we're going to pull back the curtain on how we've engineered our crops to be better, stronger, and way more reliable.
Right, so that's the big question, isn't it? It's a puzzle that plant breeders have been working on for more than 100 years, and it has huge implications for, feeding the planet. So do you go out and search the world for something new, or do you try to perfect what you've already got?
So let's kick things off with the most straightforward approach. Sometimes, the absolute best plant for what you need already exists, just somewhere else in the world. The whole strategy is about finding it and bringing it home. The official term for this is plant introduction. You can think of it like an agricultural talent scout. You're searching for a specific genotype, that's the plant's unique genetic code, that you think can make it big in a brand new place.
Okay, now this is where it gets really interesting, because this can go one of two ways. Sometimes you hit the jackpot. The plant you bring in is an instant star and ready to go. That's a primary introduction. A classic example is the Mexipac 65 wheat, which completely transformed farming in Pakistan. But other times, the new plant is more like raw talent. It's got potential, but it needs work. So you use it as a parent in a breeding program to create something even better, which is what happened with the wheat line that eventually became NIFA Bathoor 08.
So why bother? Well, the upsides are just massive. This is easily the fastest and cheapest way to get your hands on amazing new genes, maybe for disease resistance or better flavor or higher yield. It's a powerful shortcut to boosting local genetic diversity.
Of course, it's definitely not a perfect solution. A plant that loves one climate might totally fail in another, and the biggest danger by far is that you could accidentally bring in a new pest or disease. That's exactly why strict quarantine procedures are absolutely essential. They would stand between a successful crop and a potential biological disaster.
So, looking outward is one powerful strategy. But what if the real potential, the real superstar, is already growing right there in your own fields? What if it's just hidden in the crowd, waiting to be discovered? This idea brings us to a much more subtle, and you could argue an even more powerful approach.
All right, so this next part of our journey is a really cool story. It all revolves around a simple experiment with some beans, a Danish botanist, and an idea that would completely change the game for genetics and farming forever.
Picture this. It's 1903. A botanist named Wilhelm Johansen is looking at a mixed bag of princess beans. He notices they're all different sizes and weights, and he asks a really profound question. Why? Is that variation something you can actually pass down to the next generation, or is it just random chance?
His experiment was just so elegant. He picked out the very heaviest beans and the very lightest beans, and he planted their seeds in separate rows, creating these distinct family lines. He let them self-pollinate for a few generations, and then he saw something incredible. Each individual family line had become super uniform. All the big differences in weight were now between the families, not within a single family.
And this is where he has his huge aha moment. He realized that once he had one of these uniform families, what he called a pure line, trying to select for the heaviest bean from inside that line did absolutely nothing. The next generation was exactly the same. Why? Because any tiny variations left were just due to the environment, things like sun or water, not genetics. He had hit the genetic bedrock. Selection was useless here.
So this gets us to the crucial term he defined, a pure line. It's all the descendants of one single self-pollinating plant that is homozygous, meaning it's genetically pure for its traits. Basically, it means every single plant in that line is a perfect genetic clone of that first parent plant.
This work also gave us two words that are absolutely fundamental in genetics. The genotype is the plant's permanent genetic blueprint. The phenotype is what you actually see, the observable plant, which is a result of that blueprint, plus all the environmental stuff. Johansen's genius was giving breeders a clear way to finally tell the two apart.
Okay, so that's the theory, and it's brilliant. But how do you actually use this idea to make a better crop out in the real world? Well, that's where we get to the practical method, and it's called pure line selection.
The whole point of pure line selection isn't about creating something new from scratch. Nope. It's more like being a detective. It's about carefully sifting through a big mixed population of plants to find and isolate that one single, superior pure line that's already there, just hidden in the crowd.
And let me tell you, this is not a quick process. Just look at this timeline. You start in year one, picking maybe thousands of promising plants.
The next year, you plant their seeds in their own little progeny rows, and you have to be absolutely ruthless, throwing out anything that looks weak or mediocre. The best ones move on to years and years of trials until finally only one champion is left to be released as a new variety.
Let that number sink in for a second. 7 to 10 years. That's the typical commitment it takes to go from one promising plant to a reliable product for farmers. We are talking about a marathon here, not a sprint.
So, after all that work, is it worth it? Oh yeah, the payoff is huge. You get a variety that is incredibly uniform and stable. Every plant in the field acts predictably, which makes everything from farming to harvesting way more efficient. It's a really precise and powerful tool.
But, and this is a big but, there are major downsides. It takes forever, for one, and that incredible uniformity, that field of perfect genetic clones, well, that's also its greatest weakness. Since there's no genetic diversity, a single new disease or a sudden change in climate could, in theory, wipe out the entire crop. It creates a system that's powerful, but also pretty fragile.
So we've walked through the science and a bit of the history, but let's bring it all home. Why does any of this matter to you when you're just trying to decide what to make for dinner?
Well, that perfect consistency you see in the produce aisle, every banana with the same curve, every tomato the same color and size, that's not an accident. That is the direct, tangible result of applying these century-old principles of selection. It's the legacy of this science.
A variety created this way has two key features. It's homozygous, meaning its genetic code is pure and locked in, and it's homogeneous, meaning every plant out in the field is basically an identical twin of its neighbor. Purity and uniformity, that's the name of the game.
So here's the bottom line. Wilhelm Johansson's work with those beans wasn't just some dusty old experiment. It was a complete game changer. He literally handed plant breeders a scientific blueprint, a step-by-step method to systematically find the very best plant in a population and turn it into a crop we can all rely on. These methods, looking outward through introduction and looking inward through pure line selection, they're the foundation of modern agriculture. They built the food system we depend on every single day. But they have their limits, right? And that leaves us with one last big question. If these are the tools that made the crops of today, what new science, what new ideas will it take to engineer the crops of tomorrow?